Hello, and welcome to Cognitum, a show dedicated to exploring the present and future of science and technology. I'm your host, Iosif Gershteyn. Today with us is Dr. Leonardo Ferreira. Leo is an immunologist at the University of California, San Francisco, at the Tang and Bluestone Labs, and a founding fellow at the Ajax Biomedical Institute. Leo, welcome. Tell us a little bit about your work at Harvard.
I was very lucky to be at Harvard at a very exciting time for biology, when we can say we really gained full editorial control of our genome with the advent of CRISPR-Cas9.
So what is CRISPR-Cas9?
So it's this really cool system that actually came from bacteria. So somehow bacteria, when they get infected with different bacterial viruses, they can keep a record of which viruses infected them in a CRISPR locus. It's like they have a set of drawers, and each drawer will have a different viral sequence, such that if this virus is ever to try to infect this bacterium again, the bacterium has seen this viral sequence before, then can employ this CRISPR-Cas9 system to destroy it. It has two components. One of them is a protein, a nuclease. It deletes, it can cut DNA. That's the Cas9 protein. And so this protein does not just cut anywhere. It needs to be guided somehow. And it's guided by a guide RNA. So it's an RNA molecule, a ribonucleic acid, that can really bind to DNA at a very specific sequence and then induce that cutting to happen. And so this way, bacteria can keep different viruses at bay.
Mm-hmm. So it's like a find and replace system for editing DNA in a live organism.
Correct. So in humans, this is really exciting because if you imagine your genome, your DNA sequence, to be a really big book, and you want to go anywhere and find the specific sentence or whole paragraph and delete it, or maybe replace with something else. You can now do it with CRISPR-Cas9 because you have your Cas9 protein, which you can scan the DNA, and then you can design your guide RNA to go find the specific word or sentence that you want. Then once you have those two together, inside the nucleus of a living human cell, you can now change that specific snippet of text and put in anything you want, where you want.
Mm-hmm. And what's the application of this technology?
So I think there are many applications. So you can imagine, for example, the first disease to have been identified was sickle cell anemia. So sickle cell anemia is a disease that is caused by a single amino acid change. So you have a protein which is made by amino acids, and so there's a single amino acid change. And so you can go in the cells of these patients and change that DNA sequence that codes for that amino acid, such that now you have the correct protein, the hemoglobin protein. And so now your red blood cells will no longer be sickle-shaped, hence the name of the disease. They'll now have a normal shape. So this is just the beginning. This is one mutation. You can now imagine go from that to all sorts of diseases where we know the gene that causes them. And so you can now really, I feel like medicine has been focused for too long on treating symptoms of diseases. With genome editing, you can actually get at the root cause of the disease to actually change the genome of the cells that cause the disease and really correct it.
And are there a lot of diseases that have a single mutation problem, which at their very core is one mutation that we've identified and proved causality of?
There is a sizable number of monogenic diseases, as we call them. And so these diseases, as you say, they have one mutation, and if we could fix that mutation in the right cell type, then that disease would be gone. And so the next question for that would be, well, can we really deliver CRISPR-Cas9 where it's needed? So I think where the field is going next is to deliver CRISPR-Cas9 not just in cells in culture, for example, with blood. We got really good at this because everyone is going to give you some of their blood, not a piece of their brain or their kidney or their liver, but they're going to give you some blood. And we got really good at taking these blood cells out, different types of blood cells, and edit their genome and actually give them back to the patient with a corrected version. So for example, for sickle cell anemia, you could take the blood cells, correct this mutation in the hemoglobin gene, and put them back in. And so now the patient would be cured. It's a blood disease that can be cured by changing this specific sequence.
And how would you target which cells get affected by the CRISPR-Cas9?
So there's a few ways of going about this. With blood, we're very lucky because immunologists like myself, we've been obsessing over the decades to really catalog every single immune cell type there is based on a sort of a barcode of surface receptors, surface proteins.
Could you explain what is a receptor?
Yes, so a receptor is a protein on the surface of a cell. So proteins are machines that do all the work inside a cell. You have DNA, which is an instruction book, and that DNA talks to the cell via RNA. It's the message molecule. Then that message gets made into proteins. And so proteins are the machines inside the cell. Some proteins live inside the cell. Other proteins will be on the surface of the cell. And so some of them they actually use for communication, how cells talk to each other. And so receptor proteins are on the outside, and they have a ligand protein that upon binding of the ligand to the receptor protein, some signal, some decision-making process will happen.
So these receptor proteins that are on the surface of the cell are somewhat like a lock, and these ligands are like the key.
Correct.
So when the key meets the lock, then what happens?
So many things can happen. It really depends on the function of that receptor. So for immune cells, you have some receptors that they function activation. So all the cells kind of get all riled up and do their function. So for example, if you were to prick your finger, there's a whole cascade of molecules that are made. And these molecules will then bind to receptors on the surface of immune cells, which tell them there's danger, there's an infection happening, we need to get into action. And so that's one class of immune receptors. And again, these immune receptors occur in different types of immune cells. Some immune cells, we call them innate immune cells. They're the first line of defense. And they're the ones who first sense the danger, what we call danger signals, danger patterns. So if a bacterium enters in your body, there are certain conserved structures on this bacterium that our innate immune cells can sense with these receptors. They'll bind to bacterial components. And once they see those components, they pound the alarm. And so they make these molecules called cytokines. They're like protein messengers. They get released in your bloodstream. And then they go call a second set of immune cells called the adaptive immune system. So T cells and B cells. Very specific immune response, very powerful, but it takes time to be mounted. And so it's basically the interplay between these different receptors in immune cells that really gives you a full-blown immune response.
So when there is a foreign body, or when there is some kind of danger to the organism, that's when the signaling is sent out to raise the army.
Correct.
And contain the problem. Very interesting, very interesting.
And actually for us, we use these receptors, well, our body uses them to raise the army to fight infection, as you just said, but also in the laboratory, we can use these receptors as markers, as basically a barcode. So if immune, for example, a T cell will express a different set of receptors from a B cell, from a natural killer cell, from a macrophage. So we have machines called flow cytometers. So cytometry is the art of measuring cells, if you will. And so you can actually take these cells, you get like a whole blood sample, and you can stain this blood sample with antibodies coupled to fluorophores. So you'll have the antibody, which is a molecule made by B cells. We can make them in the lab. You can make them very specific to recognize a specific receptor on the surface of a cell. And then you couple this antibody to a fluorophore, a molecule that emits fluorescence when stimulated by a laser. So then, let's say I have 3 antibodies, and one will fluoresce blue, one red, one green. And then if you have a cell that emits, that will have the receptor that is bound by the antibody in blue but not in green, you can imagine a machine that could separate these cells apart. And so this is actually what I do most days. I can get a whole blood sample from a patient or from a healthy individual, and I can preparate, I can stain those cells and then purify the specific cell populations that I want based on their code of receptors on their surface.
So these receptors, which are like locks, need specific keys.
Sure.
And you can use keys to find and sort the different types of cells because different types of cells have different types of receptors.
Correct.
And that way you can differentiate them and isolate them.
Physically isolate them. Correct.
Physically isolate them. And what's the main benefit of being able to physically isolate one type of cell?
So one obvious benefit would be you can study that cell in isolation. Imagine you have a complex immune response. How do you know if the T cell is most important, if the B cell is most important? And actually I should say, within T cells there's many types of T cells. So which type of T cell is a key player in this disease phenotype, or in this vaccine, or in this specific treatment? So if you can physically isolate them, you can do assays, you can study them separately without having the other confounders. So that's a very first benefit. A second benefit would be for therapy. If you're using these cells as a therapy for a person, then you want to give them only the active agent. You don't want to have any confounding cells there. So if you can physically isolate these cells, you can use them on their own as a therapy for patients.
Mm-hmm. And what were your interests with this technology?
So when we first used this, again, it was a crazy time. It was basically around 2013. In January 2013, the first papers came out showing that you can use CRISPR-Cas9 to efficiently edit the genome of human cells in a petri dish, so cells grow in a laboratory. Later that year, CRISPR was used to make new mouse models, so rodent models for disease, and they could modify 3-4 genes at once, which was unheard of, because before making a mouse model, genetic model, would take years, and now they just did it so fast. And in 2014, actually, we were the first to use CRISPR-Cas9 in human primary cells, blood cells. So we took blood stem cells, and we took primary human T cells, and we modified their genome with CRISPR-Cas9. And in this case, we actually focused on one terrible disease, HIV. Thousands of people die of HIV every day and contract the disease every day. And what's been so hard about curing it or treating it forever is that the HIV virus mutates very fast. It's like an arms race between the immune system and the virus. And the virus has been winning every time. And that's because one of the reasons why is because the virus actually infects CD4 T cells. So the T cells that to recognize
What are CD4 T cells, and why are they important?
So, so there's two main types of T cells. Cytotoxic T cells, the killer T cells. There's CD8 T cells. So CD is a receptor. So I can stain with an antibody with a fluorophore for CD8, so I can purify CD8 T cells. And so the numbers, because we're very lazy with names, and so we just number all the receptors 1 through 300 and something by today. So you have CD8 T cells, which are killer T cells, and then CD4 T cells are helper T cells. So as the name indicates, they help immune responses to reach their full potential.
So CD8 are killer T cells and CD14 are helper T cells.
Correct, CD4.
CD4. And could you give some more explanation of what T cells themselves are?
Yes, so T cells are adaptive immune cells, so they're not at the first line of defense, they're summoned later on, and they're very special because they have two properties that they have. One thing they can do is that they can as a whole, they can kind of tell the future, because when you're born, you have almost your full repertoire of T cells. Each T cell has a different T cell receptor, TCR. And this T cell receptor can see a very specific peptide, so a piece of a protein that is presented on the surface of another cell by a billboard protein called MHC, the major histocompatibility complex, also called HLA in humans, human leukocyte antigen. And so why this is really interesting is because, so you're born with this set of cells. You can have billions of different T cell receptors, and they can help you fight an infection that you find later in life. So somehow you have enough diversity in your T cell repertoire that there will be, by chance, a T cell in your repertoire that can see a flu peptide and other sorts of peptides.
And these peptides, could you explain what they are?
Yeah, so these peptides are pieces of proteins that every cell makes. So it's inside a cell is like a constant evolving entity, and so a protein is produced, but it also has to go and be died or degraded. And so there's a structure called proteasomes. So proteasome, so some is a body from the Greek word, and so it's a body that degrades proteins. So when proteins get degraded, they'll be made into peptides. And then these peptides can be transported to the surface of the cell and presented by yet another receptor, which is the MHC, or the HLA in humans. And so this is basically a way of cells to tell their identity to immune cells. So T cells will go around your body, and they will scan your cells. And usually, it's just a normal cell. So it's making your proteins like every other cell. And your T cell has been educated in the thymus to let that go. If it's your own protein fragment, it's okay. But imagine it's a viral infected cell. So the virus somehow got inside the cell and hijacked the whole system of the cell to make its own proteins. And everything is going great so far. But now those proteins get chopped up and presented on the surface of this infected cell. So now the T cells, and once again by chance, you'll have T cells that recognize very strongly a peptide made from a viral protein that you've never seen before. But a T cell will have just a lock for that peptide key. And so this is how you can eliminate very quickly, in most cases, viral infected cells.
And how big is the universe of these potential peptides?
So those would be 10 to the 16, some absurd number like that. And in practice, it's never that big because some peptides and some receptor peptide pairs never happen due to other constraints. But in theory, you can imagine it's what's called VDJ recombination. So you have a gene locus that can be rearranged. So you have many options for V and many options for J, and then you just pick one piece of each. And then on top of that combinatorial exercise, the junction between V and J is random. There's a random nucleotide sequence in there. So this is how you can have trillions and trillions of different options. And this is why, again, no matter what virus faces you, there will be someone in the population, the human population, that will have the perfect T cell to recognize that peptide.
Someone in the population, or likely the individual himself?
So it works at both levels. So one interesting thing is that, I should say that all MHC molecules, there's many flavors of each one of them. So for example, for MHC class 1, so the one that sees killer T cells, you have HLA-A, HLA-B, HLA-C. These are different genes, but then each gene has different alleles, so sequences, flavors of that gene. And there's more than 2,000 versions of HLA-A, more than 2,000 versions of HLA-B. And so this is why, if you look at a population, there's a huge variety of HLA combinations. And so you could ask, why does evolution keep such a costly process of having many versions of one gene? And that's because, once again, if there were a plague to happen, it will never take all the population away. Because just by chance, there will be someone with an HLA molecule that is very good at presenting this viral peptide, and then has a corresponding T cell to kill those cells. So a portion of the people would always survive any plague that comes at us because of that immense diversity.
So evolution works on groups, not so much on individuals.
That seems to be the case, yes.
When it comes to immunology. And so you worked on HIV, on using CRISPR-Cas9 to interact with HIV, and we now understand that HIV outsmarts our immune system, and that's been a key problem in trying to cure it.
Correct. So our approach was actually not to focus on the HIV virus, but on our own cells. Because again, the HIV virus changes really fast. So whatever target you pick, a few months from now will have to be a different target. And actually the current therapy, which is the antiretroviral therapy, it's a cocktail of drugs that target different parts of the HIV viral cycle: replication, integration, production of viral particles. You need to take 3 or 4 or 5 drugs all at once, every day, and every once in a while, you need to change that cocktail. And so that means that it is a constant race. What if we could instead focus on our own cells that are being infected by HIV? And so it actually turns out that there's a very lucky subset of people that never get AIDS, even if they're infected with HIV. So these people, we found that a mutation in a specific gene called CCR5. CCR5 is a receptor expressed on the CD4 T cells that HIV absolutely requires to infect these cells. So if we take away the lock, now HIV can no longer enter the cell. It's like we're shutting the door on HIV.
And how did you find this CCR5 receptor deficient people and the use of this receptor? Because there are so many receptors.
So this is from a very cool story that actually came from Germany. So this American man that he actually studied abroad in Germany, and he contracted HIV while he was there. And then later on, he actually also was diagnosed with leukemia. So he needed a bone marrow transplant to cure him of his cancer. And the German doctors, they found someone that's compatible with him, genetically matched to him at the HLA level. So you not have any problem. And so what happened there is that the person that was compatible with him was one of those people that won the genetic lottery, if you will. This person had a mutation in CCR5, such that this person would never contract AIDS. And so when he got this matched bone marrow transplant from this individual, he was cured from HIV. And it's now been over 10 years, and he's still HIV free. So that was our case. This was Timothy Ray Brown. That's the name of the individual. And so we knew from that one-time experiment that almost nature and medicine made that you can cure HIV by introducing cells that are resistant to HIV. They just cannot be infected by HIV.
So HIV requires the CCR5 receptor.
95% of HIV variants do. I should say there's another variant that actually uses CXCR4, a different receptor. But you can imagine with our approach, we can go in, we can give you some drugs that will recruit blood stem cells from the bone marrow into your circulating blood. We take a blood sample and you can purify the blood stem cells using flow cytometry, and then you can purify those cells, you can modify their genome, you can modify their genome with CRISPR-Cas9 to delete this gene. It is an RNA that will bind to CCR5 gene, and then Cas9 will go and cut that DNA there, and then just the way cells repair DNA cuts, there's two ways they do it. One way that they do it is that they actually do a really good job, and it looks just like before, and that's called homology-directed repair. So there's homology just means the sequence looks just like what was there before. And so when cells are cycling, they have two pairs of chromosomes, so they have kind of a template to repair from. But most of the times, if you just induce a double strand break, the cells will just stitch it together to prevent them from dying. Okay, if cells are just open breaks, they'll just die. And so when they stitch it back together by non-homology end joining, they just stitch together the ends. And every time they do this, or many times they do this, they insert mistakes. They leave a base pair out, or they put another base pair in. And so what this effectively does is that it knocks out expression from that gene, because now the sequence is wrong. You can imagine if you have a system where each 3 letters in a genome will give rise to a codon, which is an amino acid, and you make that protein. But if you now were to mess it up in 1 letter, now everything is out of frame. We literally use the word frame because that's where it is. So when this sequence is now read, because it's out of frame, the whole protein is messed up. And there's even like a premature stop codon, so it just tells the whole protein production to stop. And so this is how we can very specifically delete expression of this gene in blood stem cells.
But does it impact the next frame? Does it impact the index? So if I rip out a page of the 50th page of a 100-page book, the first 49 pages, I know, oh, I need to read the 33rd page. I go to the 33rd page. But when I say, oh, I need to read the 55th page, I'm actually reading the 56th.
So luckily, genes are not all next to each other in our genome. So even if you mess up the frame in one gene, that does not alter the frame in other genes. So you can specifically delete this gene, its expression, without affecting any other gene. Because our genome is actually, if you take our whole genome, less than 2% of our base pairs in our genome, which are 3 billion of them, only 2% of those 3 billion base pairs actually code for protein coding genes. So there's a lot of space between different genes.
So now that we can edit the genome and we know the receptors that are allowing us to be infected by this horrible, horrible disease, is the end of HIV in sight?
We would hope so. I guess one thing is that we need to first make sure that this receptor that we're targeting has no other important function in the normal individual. So that the fact that there's people walking around that they don't express this receptor, their cells just don't have this receptor, that gives us some confidence, some hope that we can live without this receptor, and that in that case, the benefits largely outweigh whatever detrimental effect that could be. Well, one question would be then, do you want to just take everyone and edit this gene? Or do you want to only use it in case of disease? And so there's actually two camps. So if you go, for example, to China, they have edited CCR5, the same exact gene, in human embryos. And so you would imagine the goal there is to, such that the next generation of babies will be resistant to HIV, or at least to most types of HIV. But then what we have to worry is that, again, the virus keeps evolving. So what if everyone didn't have the receptor? Then maybe the virus would find another receptor to enter. And as I mentioned, a small percentage of HIV variants actually uses CXCR4, not CCR5. So I think actually with all gene therapy approaches, they should still be used with some caution, which is that they should be used to treat disease and not necessarily to alter a trait in embryos.
And as a treatment for disease, what would be your best estimate of when we would see this being applied therapeutically?
So right now we licensed our intellectual property to a company in Waltham, and there's some clinical trials happening in China. And so I think the first step is always safety, to make sure that these cells are not modified in some other way that will be detrimental. From our own preclinical studies, it seems that if you modify this gene in blood stem cells, they maintain all their properties. They can give rise to all blood cell types, they grow at the same speed, they don't develop cancer at any high rate. So that way, and again, and from the case of Timothy Ray Brown, it seems to be a safe approach. That will be the first step. The second step is it enough to make your cells resistant to HIV infection after you've been infected? Because one argument for that idea is that these blood stem cells can basically repopulate, they can replace your whole immune system. Because the blood stem cell, which is the first type of stem cell to be discovered, it can renew indefinitely, and it can give rise to all cell types in your blood. So as your T cells are slowly dying, your T cell counts are like close to zero in HIV patients, once you put these HSCs, these blood stem cells in, these cells don't care about HIV, they can be infected by it. So they can differentiate into all blood cell types, they can replace your pool of T cells. And more than that, these T cells, some of them will recognize HIV peptides. So they can go and they can kill HIV-infected cells. So that's the good news. The not so good news is that actually HIV was found to exist in some reservoirs. So there are some cells that they have HIV inside them, but they actually don't make HIV. So it's like kind of this like backup plan for HIV to stay inside your body. So there's a bunch of treatments trying to reactivate that gene, that HIV gene machinery, to then make the cells visible to the immune system. That's something that is currently the challenge in the field. Can we eliminate all HIV? Do we need to eliminate all HIV in the body? We don't know.
Well, we hope you'll find out. Thank you for joining us this evening. We hope you join us again next time, where we explore the frontiers of science and technology. Have a good evening.